Good evening and welcome to all students, staff, faculty, invited guests who have gathered here to felicitate Professor Jayant Udgaonkar, Faculty of Biology and our ex-director for receiving the Vigyan Shri Award for the year 2024. This award is part of the new Rashtriya Vigyan Puraskar Awards initiated by the Government of India to honor exceptional contributions to the fields of science and technology. Now I'll request Professor Girish Ratnaparki to come on stage and give a brief overview of Professor Udgaonkar's contributions to science and significance of the Vigyan Shri Awards.
Thank you, Marika. I have a few slides. So Pinaki asked me to give a short introduction before Jayant told us about his career and his career's work. There were two reasons, I guess, why I am giving an introduction to Jayant. The first reason is that around the time Jayant started his lab in NCBS, almost, I guess, 35 years ago, I was in the neighborhood. I was a humble graduate student at that point. And I had interacted with Jayant's group, and I have actually one publication with Jayant. So that is one of the reasons. And what I'm going to focus on is basically talking about that era, which is mostly the 1990s. The second reason is that, again, in that time, I worked on the problem which Jayant has focused on for most of his career, which is the protein folding problem. So two reasons. And of course, I'm, as always, very happy to be here and be part of honoring Jayant. So let's start off. It's about then and now. And for students, there are not too many over here. The Gen Z students who were born, let's say, after the year 2000. This is Jayant in those days. The world was black and white then. Jayant was in color, of course, but everything was more or less in black and white. You can see the young Jayant, and you can see not much has changed. He had a fancy computer, state of the art, most expensive you could buy, and it had only 256 shades of gray. That's pretty much you could see over there. To emphasize on how long ago it was, here are three major events happening that year, 1990. The German wall had been broken. 1990 was the year of reunification, really a long time ago. Madonna, for those who know who Madonna is, basically put out her song Vogue, which you can hear in YouTube even today. And Friends, I realized Gen Z still watches Friends, has its first season. So ancient times, last century, a long time ago. That's when Jayant started his lab. So Jayant, for almost all his career, worked on the protein folding problem. And this was a very nice cartoon, and I'll show you another one in a few slides, made for the Trends in Biochemical Sciences, which was a review journal, by a person called T.A. Bramley. Now, T.A. Bramley never revealed his name. I had to spend a lot of time yesterday night to figure out who Tab was. He would just sign himself off as drawn for tips by Tab. Okay, so Tab in the year 1991 made this very beautiful cartoon, which will give you an idea of what Jayant's life work is. You can see the ribosome on the left-hand corner, and if I'm lucky, I'll be able to show no, I can't do that. You can see the amino acid chain coming off the ribosome, and in the cell, in the crowded cell, which is being depicted as a jungle over here, you can see that the N-terminus of the polypeptide chain, as it's trying to fold, which is the interest of many people at that time and even today. There are all these other accessory proteins shown as nannies over here, which are helping out the protein to fold. And they are making sure that the protein doesn't misfold, because the folded state is what more or less dictates proper functioning of the protein. And Jayant didn't spend much of his time doing his work in vivo. He did most of his work in vivo, which is on the right-hand side. And you can see that small proteins, the kind of proteins which Jayant worked on, when left alone in solution in a decent buffer, would happily fold by themselves. And this was the Nobel Prize for Christian Anfinsen, way back in the early 1900s, with the idea that a sequence of amino acids can fold by itself without any other information. Though, of course, in vivo, the situation is very different. This is what Jayant's lab looked like in the 1990s. you can see two of his students who are not young anymore, which is Ramya and Utpal many years ago. Jayant's lab, if you walked in, was pretty famous for its top flow machine, which he would use to measure folding of a protein. If I remember right, in the millisecond domain at that point, I'm sure things have moved forward from that point. The lab had spectrophotometers, CD spectroscopy, a fluorimeter, and Jayant's lab would monitor the folding of a protein starting from a denatured state. And another major experiment, which I'm sure Jayant will talk about, is doing hydrogen exchange either with NMR or with mass spec or CD to look at the collapse and folding of proteins. My link with Jayant was this protein, which is shown on the right-hand side with the PDB ID 1A19. I'm very proud to tell you that I solved this structure. And our labs collaborated to work on the structure of the uncomplex state of bar star. And again, a very nice cartoon from TAB showing bar star, which is basically an inhibitor for a ribonuclease called barnase. And the structure we solved was actually a mutant variant of bar star, which is kind of depicted in this cartoon. Both barnase, the ribonuclease, and bar star, its inhibitor, are from Bacillus emoli liquefaciens, and regulation of RNase activity is very critical. And Jayant spent, I think, most of the 1990s working on bar star. This is where Jayant was. I call it the land of NCBS. It's a far away place in space and time, very special place. And this picture was taken in the 1990s, when NCBS was a much smaller place than it is today. I have put an arrow to point out where Jayant was, a much younger Jayant at that point. And for my colleagues who are in my age group, if you pay attention, you can see a lot of very, very important people, especially on the left-hand corner. If you pay attention, you will see a very important person as far as IISER is concerned, and I won't talk about it anymore. This photo also holds relevance to me, and those of you who are getting what I'm saying understand what I'm saying, and I will say no more about it. So NCBS in the early 1990s started off in the campus of the Indian Institute of Science. And Jayant was where you see the red star on the top somewhere, which is pretty much where NCBS was started. And all the new faculty joined NCBS in that new campus. And by 1999, they shifted to the GKVK campus, and which is where NCBS is today. And Jayant, in the 1990s, early part of his career, spent his time in both these places. So I'll stop this introduction, which is basically about then and now. You can see how Jayant has evolved, let us say, from his early years to today. Many years have passed. He has mentored many, many students. He has a large bunch of papers, excellent papers on protein folding. He has been, he was a dean at NCBS. He was director of IISER Pune. He has many awards, which I will not name. What I've done in the bottom part of this slide is to just do a text search on words used in the title of his papers. As you can see, folding is almost 52 out of his 150 papers. If you look at the abstract, I'm sure it will go up to 150. But a large number of his papers are about on folding. Bar star, which dominated his life, I think it dominated his life in the 1990s, has a large number of hits. Other interesting words which he will introduce you are intermediate, amyloid, native state, dry and wet molten globule, God knows what that is, and collapse. So these are the keywords which you will hear. Thank you very much. Congratulations, Jayant.
Thank you, Girish, for your lovely presentation of Jayant's life from 35 years ago till today. So now I'd like to call upon Professor Sunil Bhagwat, our director, to come and say a few words on this occasion.
Professor Jayant, Mrs. Udgaonkar, colleagues, students, very good afternoon to you all. My first encounter with Jayant was a few years ago, but it was through one of those computer screens. If you remember, it was COVID times, and we were conducting interviews online. And that's how I learned about how he operates as a director at that time. His work on protein folding, Girish has explained to me as a chemistry person, proteins are molecules, but they are not just molecules by themselves unless they fold, unfold, aggregate, and they catalyze things. They do a lot of things which is not possible by just knowing their chemical structure. Besides that, I think Girish was there to explain. But as a director, when he worked here, I could pick up a lot of things what to do when I came here through interactions with him. And contrary to my initial apprehension, how his guidance would be for me working here, he has been a wonderful colleague and a prior director who has always been available for any inputs that I may require. And Girish, you did find one photograph with his playing tennis, right? Yeah, okay. So he's a tennis player. And all of you who have been colleagues with him for a longer duration than I am know him for his work as well as his leadership. And these awards usually come a little later compared to the work you do. He was recipient of the Shanti Sarup Bhatnagar Award, which is given before 45 years of age in 2000. The Vigyan Shri is in the series of 3 new awards which have been started by the government. Vigyan Shri Yuva, which is equivalent to the Bhatnagar Award. Vigyan Shri, which Jayant, and glad to tell you 3 more IISERites, not from here, that is 2 more IISERites from IISER Bhopal are also in the category of Vigyan Shri this year. And Vigyan Ratna, which is either 1 or 2 at the most. So it's the next award which Jayant may give us opportunity to celebrate is also known. So Vigyan Shri is a very prestigious award given not just for biology or chemistry, it's given for any of the Vigyan related works. So definitely a very prestigious achievement. Once again, congratulations and hope to see more from you soon. Thank you.
Thank you, Professor Bhagwat. Now I'd like to request Dr. Nathu to come on stage and say a few words.
Dr. and Mrs. Udgaonkar and my dear colleagues. About Jayant, everybody has talked about protein folding and all other things. Talking about protein folding by me is like taking a cold to the Newcastle. So certainly I will not do that. I don't dare to do that. But when this new award started, as director has already said, they wanted to integrate all the awards into one stream, as this is done in case of civilian awards on 26 January, like Padma Bhushan. Slowly all this will come. Right now only Vigyan Ratna and Vigyan Shri has been given, but there may be some others from next year onwards. Why this way? Why it is not called Vigyan, and what is Shri about it? So when I went to the dictionary and the real meaning of Shri, what happens if you add Shri to Vigyan? Vigyan is Jayant's expert and everything. But why Shri? Then I realized Shri means several things which I was not aware. Number one, it is a title for wisdom. In fact, then I realized whether the Shri's meaning have been taken after looking at Jayant. One thing, it is a title for wisdom. Nobody has to say anything, and he has been duly awarded for all these things. Second, wealth is not wealth, pure wealth. It is wealth of virtue by virtue of excellence. That means it is not anybody money obtained from trade or anything, but is virtue of excellence. So all these qualities exactly fit into Jayant's matters. Third was it spreads knowledge, which is very true. You have seen the number of PhD students and his talks and all other things. Then it is beauty in concentration, which is also there. Without that, I don't think he'll be able to, he has progressed. And besides all these things, I consider these to be all adjectives of a fine gentleman, and that to fine, transparent gentleman. So I feel Jayant deserves all these things, and I would like to pay my personal tributes to him on this occasion. Besides protein folding, he has three other interests. Number one, or three other roles rather to play, and that also he has excelled, and that in number one is administration. He has in these 5 years, he has proven to be a good leader, and good academicians, this is a very rare combination that he has been proved to be. Second thing is very close to the family because he has gone up. So Mrs. Udgaonkar is there, so she also deserves compliment to supplement him or compliment him. I really appreciate that. Third thing is, well said, is sports. His love for the sports, especially of tennis, is a very, very, has been seen or watched by many people in IISER. So thus, a man who is academic leader, excellent scientist, able administrator, and a good transparent human being. These are the four qualities which I'll assign to. And these are the qualities of the word Shri. So Vigyan is a science and Shri describes him in person. So this is how I feel this is a unique combination. And I really salute to him on this occasion of Vigyan Shri. And as he said, I hope and we all hope that he'll continue to get these kind of new hats and which we'll be again celebrating in the same hall. I hope that opportunity will come soon. Thank you very much.
Thank you, Dr. Nathu. And now I'll call upon Suman Pal, a graduate student in Jayant's lab, to come and say a few words about Jayant and maybe how life is in the Udgaonkar lab.
Okay, so good evening, and firstly I'd like to thank the organizers for giving me this opportunity. Indeed, this is a very proud moment for all of us, and I have been working with Jayant for almost 6 years, and it's been a wonderful experience. And if I want to share something about his mentoring style, the thing I would like to point out is respectful and non-condescending approach towards the student, which allowed the students to be unafraid to ask anything, and which I found is very rewarding because the students take full ownership and the responsibilities of the project, and that's a real confidence booster in the initial stages of the PhDs. He also taught us to think critically, not only about science and also about life. He also encourages us to focus, to pay attention on the details. He also tells us that we should have a sane balance between our personal and professional commitments without compromising either. So he does have a strong affection towards the instruments we have in the lab. He refer them as his babies, and he does make sure the students take care of the instrument. And he has been extremely tolerant and patient, although there are multiple times we have tested his tolerant limit. And on a personal note, he has been a very cheerful presence in the lab, and he has always lightened up the mood with playful sarcasm and his jokes. Some of them are really good, some of them are not so good, but that does the job. And he does have a good sense of humor. Yeah, so yeah, congratulations, Jayant. And I'm really, this has been a really enriching experience for me to having a supervisor like him. Thank you.
Thank you, Suman. Now I'd like to request Professor Jayant Udgaonkar to come on stage and share his experiences, challenges, and achievements in his scientific career.
Well, I'd like to begin by thanking Sunil and the Institute for this felicitation. I get embarrassed by felicitations because that's the type of person I am. But on the other hand, felicitation is in time to acknowledge the contributions of many people, the contributions of students in particular, collaborators, colleagues with whom one has had discussions, support staff, everyone who keeps an institute such as this or NCBS running well. And so thank you all for that. So it's difficult to give a 30-minute talk on one's journey, and difficult because you don't know what one should say and what one should not say. So, but any journey begins from the time one appears on earth. And so I come from a family with an academic background. My mother was a PhD in history, a historian. My father was a theoretical physicist. My mother's father was a professor in Banaras in the university. And so coming from an academic background, one realizes the importance of really scholarship in doing well. And for that, really, I have to acknowledge what my parents contributed. I spent my first 4 years in the two top universities in the US, but as you can see, I wasn't doing science at that time. I was a little too young for that. So I had my schooling in Bombay, Mumbai now. I was an average student, didn't do well, in spite of having absolutely excellent teachers. Our teachers really taught us to think, taught us to ask questions, taught us to challenge them. And I think that really played an important role in my future scientific development. Um, so I graduated with average grades. I should say that my class was well above average, so it's not too bad being average in that class. Out of 24 science students, I think 14 or 15 went into IIT. I got admission, but I chose not to because I wanted to do biology. Uh, so what was somewhat important for me was that in the final year of my school, I learned electronics. And that was very important for me because both in my PhD and in my postdoctoral work, I had to build things. And this experience where I built radios at this time with Mr. Kalia at TIFA really had set the stage for me not to be afraid to do such things. So, so that was my early years in school. It was a very good school, as I said. I then went on to do my BSc in chemistry. I said I was interested in biology, but in college we had absolutely fantastic teachers in chemistry. And so I got very interested in chemistry, especially physical organic chemistry. And It was a good time. St. Xavier's College is a college which had very good teachers, and I really enjoyed myself there. But what was transformative for me was that in the summer of 1978, when I was in the second year of my BSc, I got the opportunity to work with Professor Maitra at TIFR. Maitra had a tremendous influence on me. He was a professor, full professor, who worked in the lab himself. And he really taught me many things about how to do science, how to think about science, and in general about life. He also taught me to smoke cigarettes. Luckily, he didn't persist when I stopped at cigarette number 2. But anyway, it was a great time, and I met many people there. I'll talk about that later. So when I was doing my BSc, finishing my BSc, decided I would do my master's in chemistry, and I applied to various IITs. All IITs had this requirement that one has to do mathematics in your BSc, and I hadn't done mathematics. So I applied to IIT Kanpur, they didn't bother to reply. IIT Bombay first said yes. When I went and argued that I had gotten through the JE and I had studied maths on my own, they said okay. But then afterwards they changed their mind because many people went and made similar requests. And then IIT Madras also had the same rule, but they seemed to have missed the fact that I hadn't done any maths, and they gave me admission when I went to get admitted. Fortunately, they didn't notice that I hadn't done maths. Of course, it was difficult doing an MSc in chemistry without having done the maths coursework, but they had 2 maths courses all chemistry students had to do, and that really turned out to be very useful. So IIT was a real difference experience. Going from a college like St. Xavier's to IIT Madras was a very strange experience for me. For the first time in my life, I was asked what my caste was. Nobody in Xavier's bothered to really ask such questions. I was the only person north of the Vindhyas, I guess, in both amongst the students as well as the faculty in the chemistry department, and I was looked upon as a strange object. So I don't know whether you know that in the South, in many southern universities, people get 95, 98% in their BSc. And I went from Bombay University with 70% or so. And so they would laugh at my marks, but finally I made good friends, and it was a really good experience. I learned to become quite good at bridge because we would start playing on a Friday evening and end on Monday morning, with of course breaks for eating and all that. So I became an expert at playing bridge. Of course, it told on my grades, so I was again an average student. But fortunately, in this exam called GRE, which one has to do to get into a PhD program, I managed to do quite well because that was in the summer holidays when I was at home and I managed to do some studies. So I should tell you, so the class topper was well above the rest of us, and she did very well in the GRE. And at that time everything came by post, and I didn't get my letter saying what my score was in the GRE. And she kept telling everybody in my class that, I'd done badly and compared to her, so that's why I'm not saying what my score was. Then finally my scorecard came. They put the incomplete address, so it had traveled a lot. And it turned out I did a little better than her. And her face really went down after that, and she didn't talk to me after that. But anyway, it was a good time in IIT. I also applied for a job in industry when I was there. And I thought the interview went very well. This was a job with Glaxo. And they asked me all sorts of questions not related to science. But finally, the person said, you are this one's son, and you're not going to take a non-academic career, so we're not going to offer you a job. So there are advantages in being the son of a well-known faculty member, but there are also disadvantages. And I don't know how someone in Glaxo knew about him. Anyway, I then went to do my PhD at Cornell, and I did in vivo biology. We worked with fish. This is a torpedo, Californica. The reason why we worked, so this was a lab which did neurochemistry. We were interested in how neuroreceptors dealt with signals coming from nerves. So the acetylcholine receptor is one of the very important neuroreceptors which function at nerve-muscle junctions and also at nerve-nerve junctions in the brain. And this particular fish and also the electric eel have specialized muscle cells with a very large number of these protein molecules. And that's how they generate few hundred volts, which they use for communication. And so that's me holding a live torpedo. It had been dumped on ice, so it becomes very quiet. And then you have to kill it to get the organ. You do that by taking a screwdriver and a hammer and hit the brain. But and that's my advisor. I purposely shown his photo because he was really someone who I think was the ideal mentor for any student. And I think I learned a lot about how to be a mentor from George. And I had a great time doing my PhD. I had to learn how to learn programming to quite a bit extent. I spent 6 months of my PhD doing programming because we were doing these measurements for which no commercial software was then available. So I was doing what was called single channel measurements on a home-built amplifier. And so my electronics, which I'd learned, turned out to be very useful. I also did an electronics course at Cornell. And so I had to write software to analyze such data. And so this took 6 months. So I had my program ready, and I decided to generate random synthetic data to check the program on. Now, as you can see, this program, the actual data has got noise in the baseline. But the synthetic data I created, I forgot to put any random noise. And I forgot that I had written the program to find the baseline by looking for noise and then getting the average of the noise. And so now I had a program which was running for several days, trying to find a baseline. Without finding the baseline, I couldn't do anything. And it went on and on and on, and my advisor got a very large computer bill because of my not being able to create good data. But anyway, so I learned a lot. This is when I learned to do chemical kinetics, both at the single molecule level as well as at the ensemble level. So then I went on for my postdoc to Stanford and into a completely different field. This was a project I heard from George told me about it, and it really excited me, the methodology which was being suggested, which hopefully would give information on protein folding with atomic resolution. And so, of course, the work involved developing the methodology, which involved fast mixing, hydrogen exchange, and an NMR, and of which I only knew fast mixing. I didn't know the other two at all. But I learned new things. It so happened that when I was at Cornell, Kurt Wuttrich came and gave his famous Baker lectures on NMR. This was the early days of biomolecular NMR. And he had these series of talks where he had two projectors, and he was a very imperious man. He said, one thumb with my whatever he is holding means left projector advances, two thumbs means the right projector advances. That's the only exposure I had to NMR at that time. But I learned it properly when I went to Stanford. And for 6 months, I was just sitting on a chair and not doing any experiments, and everyone around me was wondering what I was up to. But finally, I picked up a pipette pen. started doing some work. So anyway, this is what was good about Stanford was the postdoctoral peers were of absolutely great quality. And that's very important while choosing a postdoc because, these are the people who are going to do well, and you get to know what type of people will be able to do well. And I made very good friends there. There's Richard and Christine and Heidi out there, and Dave, and David Schultz out here, and Andy Robertson. And we really had good times at Stanford, both in the lab and outside. So towards the end, I had to make this decision about whether to come back to India for a job or continue in the US. I was under a lot of pressure to remain in the US. And that was actually quite stressful. I had, right from the beginning, decided that I would come back to India. But when the time actually came, because of the pressure of various people, it became pretty stressful, especially when my postdoctoral advisor more or less secured me a faculty position at a pretty good university. But anyway, I did come back. I had applied to Indian Institute of Science, to the molecular biology unit. They didn't reply, and then Professor Balram, for some reason, wrote to me and said, you applied to the chemistry department, the IPC department. So I did, and they offered me a job. And I'd also applied to this new center at TIFR, which I'd heard about, and to TIFR proper. TIFR proper didn't reply, but Obaid Siddiqui, who was in charge of the new center, did reply. And I was offered a job there. So I came back to India in October '89, and for 3 months I couldn't decide where to join. I visited ISE, met CNR Rao. Since I was being put in a chemistry department, I was quite worried about having the necessary biology equipment, so I said I need so much money for that. It was a small amount by today's amounts. And he said that he would write to me confirming that. So I was waiting and waiting in Bombay for his letter to come. But he wrote to my previous address at Stanford. I'd already left there. And of course, it took several months for that letter to be redirected to me. In the meantime, I met Vijay Raghavan. And Vijay has this knack of talking with great confidence and has great people skills. And by that time, I had already been inclined towards joining TIFR because I decided that the chemistry part of my work was not so much of a problem for me as the biology part. And so I decided the biology group at TIFR would be a good place to start off at. And then Vijay, of course, then convinced me that I should. So I joined NCBS, or at that time it was being called a Center for Biology, being set up and in Bangalore. So I joined them. I was just below 30 years old when I joined. And after joining, I realized that the center had not been approved by the government. And so that means no money for it. And also that the location in Bangalore, as being in Bangalore, was not yet approved. So Vijay and I made a trip to Pune in 1990 because the government was asking, why not in Pune? So we came to Pune. The government arranged for 2 sites to be visited, large tracts of land which were well outside Pune at that time, but now I think are well within the city limits. And we came back, went back to Bombay and wrote a letter saying why those 2 parcels of land were not good enough for NCBS. And it's ironic that I ended up in Pune at the end of my career. But anyway, so we finally got the approvals 2 years after I joined, and everything got set and things worked out quite well. But in the first few years, I worked in, so let me first now tell you what I started working on. The problem I was working on is what's called the protein folding problem. So you have a necklace, a polypeptide chain of beads of 20 different colors. And unlike a necklace which will just flip-flop around, a protein chain adopts a very well-defined structure where positions of atoms are precise to within fractions of an angstrom. And that precision is important for the function of the protein. So the folding process is what allows this polypeptide chain to adopt this very intricate structure. This is a small protein which Girish introduced you to. And you have beads of 20 different colors, or what are called amino acid residues of different, with letters of different letters of the alphabet, giving you this structure. And the folding problem is how do you achieve this structure from this sequence? So this structure is very important. So I'll just give a small introduction to the, can make out some red colored and blue colored objects on the left and right. So these are two different enzymes, one found inside your gut that allows you to digest food. The other is found in a bacterium, and its famous subtlysin is found in a bacterium, and it's the, so you have this, so you see two proteins, you can make out that the structures are very different. Both have the same function, both chew up other proteins. You can see that the part which is active, which allows the chewing up, which does the chewing up, are shown out here and out here. And you can see that the configuration of the atoms in this part and this part are virtually identical. And because these atoms are placed in the same orientation with respect to each other, the same distances apart, these proteins can chew up other proteins. This is found in your gut, allows you to digest your food. This is found in bacteria, where it's used as defensive mechanisms. But this protein was made famous by Genentech, because Genentech decided that it could be used in laundry detergent. Those of you who use Tide would be cleaning their clothes with detergent containing this particular enzyme. And what Genentech did by brute force was to make this protein capable of functioning in at high temperature and in alkali conditions. So folding is something which allows that, it's going the wrong way. Which allows this structure to be attained. You can see that you have two completely different scaffolds, but both scaffolds allow the arrangement of the active atoms in the protein molecule. So folding is happening all the time inside your cell. It's not just when proteins are synthesized. Because proteins are marginally stable, proteins are folding, unfolding all the time. And in certain types of activities, for example, anytime you move even a little bit, a protein is folding and unfolding. So for example, when muscles contract and relax, you have two proteins, myosin and actin, which go like this. And if you have two proteins going like this, you don't want them to get out of register. So nature uses a rubber band, which is another protein, and that protein functions as a rubber band by having regions in it which fold and unfold. So when they unfold, you're extending your rubber band. When you fold, your rubber band is contracting. So this is happening all the time inside you. So there are two facets to the problem. One facet has basically been solved recently because of developments in AI. So the program AlphaFold really now allows the structures of proteins to be predicted, but they don't tell you how and why a particular structure is achieved. And that's what my lab has been working on for the last 35 or so years. So AlphaFold is very good in predicting structures, but it doesn't tell you anything about how the structures are reached. So at the time when I started my lab, there were various broad ideas about how proteins might fold. We had different models, maybe secondary structures, helices, etc., form first, and then you had collapse of all these structures to get you some intermediate state and then giving you the folded protein, or the chain collapse and you arrived to the native state, or you had models such as this which came a little later, which allowed the native structure to be reached. At the same time, or roughly a little later, the statistical mechanical models of folding arrived on the scene, which for the first time, experimentalists started looking at these objects not as discrete structures, but as ensembles of molecules of different structures. And the idea was that folding landscapes were funnel shaped and molecules would come down the slopes just like a skier comes down a ski slope, or from the top of the mountains, they're coming down to the plains. You can do it in many different ways. And so the idea was that individual molecules were all doing it in many different ways. So these were somewhat extreme views at that time. And that's when we started working on the. So I joined TIFR since the center didn't exist. I spent the first 3 years at TIFR Bombay, and that's me with two colleagues who joined within a year of my joining, Mani Ramaswamy and Matthew. This photo Girish already showed you. We had a good time then. I was setting up my lab. I spent a lot of time setting up my lab. And we had good times both in the lab and outside the lab. So Matthew remained at NCBS. Mani left after 2 years. But having them around was really something, especially having Matthew around for 30 odd years, really enriched my science and my thinking about science. So then I moved to the TIFR Center and the IISc campus, and Girish has already shown you these photographs. And so Utpal, as many of you know, is now a professor at IISc. And this was a good time. We were setting up things. We were small groups. At IISc, we didn't grow to more than 10 faculty members. And we worked together quite well. Unlike at IISER, we started off with very little money. NCBS now looks very rich, but at the beginning, we didn't have much money. And I remember when 3 of us got big grants, I told the other 2 that we are not taking any money from NCBS because we need it for the new faculty members. And so the 3 of us survived on our grants for several years before, and by that time, NCBS started getting money. So it was a good time. We shared equipment, shared ideas, shared everything. And the whole of NCBS had one centrifuge. Every lab in IISc had its own centrifuge. But things worked out, and it was good. So for 5 years, I was at the TIFR Center, and then my lab was the first to move to the new campus in 1998. The move went quite well, except for a couple of months, my students would have to come back to the TIFR Center for doing things like adjusting pH. Because we had one pH meter, the people left back didn't want that pH meter to move without them. So it was a little difficult, but anyway, things worked well. And over the years, I spent nearly 20 years on the NCBS campus there. And I'll talk about lab work and other work in a while, but it was a very good environment which I had the lucky chance to really shape in the years to come. So, and then 2017, I moved here, my lab moved here. I couldn't move here without having my lab next to me. So my lab has moved from 1, 2, 3, 4 locations, and it's been good every time. And fortunately, nothing broke in any of the 3 moves which were involved. So on the right is my current lab. This was the lab quite soon after. On the left is the labs very soon after I joined. So I should tell you a little about the science. So the folding problem, as I told you, is how does this polypeptide chain, which can diffuse in all possible directions, adopt this very precise structure. And we were, as experimentalists, we were asking questions very fundamental in nature. How does the process begin? What happens first? What happens next? Is it a diffusive process? Are there discrete states being formed along the way? And these were questions which were important to the field. Some of them are still unanswered now. And questions which we have addressed to some extent or the other over the years. And I'll talk about that in a bit. So before I talk about those questions, I'll talk about this paper. because Girish told me he likes this paper a lot. It's by his friend Vishwas Agashe. And, but not because of that. It's thermodynamics, which Girish used to do at that time. And this was, to me, an important paper because it really was the first time anybody had characterized what's called cold denaturation. We all know proteins unfold when you heat them. We've all perhaps boiled eggs. But they also unfold when you cool them. But nobody had understood what happens to the protein chain during this process of cold denaturation. And because luckily I had visited Stanford for 3 months in 2013 and used the CD machine out there. And, because those people did peptide work, they used to work at low temperatures, so the temperature scans would start at 1 degree and go up. And for proteins, normally one would start at room temperature and go up. But because it started at 1 degree, I noticed that the protein was getting unfolded at the low temperature. And so when I came back, Vishwas characterized this whole process in great detail. And that really allowed us to understand a lot about how proteins unfold and the thermodynamics of protein unfolding. We didn't realize many things at that time, and about 20 years later or 18 years later, we did similar work on the prion protein. And at that time, I had matured more as a scientist, understood more about proteins, and we use the measurements we made to show that the hydrophobic effect is not the main force stabilizing the interaction. Other things, hydration of polar surfaces and packing interactions are equally, if not more important, along with the hydrophobic interactions. So the hydrophobic interactions are interactions involving non-polar regions of the protein coming together. So we started working on protein chain collapse, and we could show that there was no structure being formed first, just the chain just collapsed. Then you started forming some structure, and then you finally ended up to the native state. So this was very early on, and we continued working on this for many years. And we have followed this up right up to perhaps 2021 was the last paper on chain collapse. And we have discovered many different ways of thinking about how chain collapse is related to protein folding. I'm not going to talk about this in detail, but it's something which really opened a new avenue of research in the lab, and we went after our discovery for many years, right up till very recently. So we then started, I'm not really going to talk in a chronological order, but I'm not sure in what order. But one of the things, in the early 90s, I started a collaboration with G. Krishnamurthy at TIFR in the chemistry group at TIFR. And that continued for 25 odd years. And we have many publications together. And one of the papers we worked with, which we did in the 90s, had indicated to us that folding is a gradual, continuous process. You don't just hop from one structure to the other. You know, the ideas in protein folding had come from small molecule chemistry, that you have one structure, it jumps over an energy barrier to another structure, then it jumps again, etc., etc. What we could show, GK and I, in the 90s, was the structure was perhaps changing in a continuous manner. And it's with this publication that we could really show that this was the case, that the native state of a protein, I'm not going to describe the methodologies because I just don't have the time for that. The molecule was expanding during the unfolding process in a gradual manner. You can see this distribution of distances changing as we kept adding urea. And so it went through a series of native-like forms, and then through a first-order transition, went into a series of unfolded-like forms. So you have a molecule swelling. Then once it reaches some critical dimension, it undergoes a first-order transition to the unfolded state and then swells again. Girish says he doesn't understand dry and wet molten globules, so this is purely Girish, pay attention. So this is, the question was, how does a protein begin to unfold? And two Russian physicists, both very well known, had proposed this dry molten globule hypothesis, where they first said the protein swells a bit, and that's when structure starts changing. Now, nobody had been able to really show that in a conclusive manner. And Santosh Jha, who's now an NCL, could show by doing distance measurements that the protein does swell first to a dry molten globule, which doesn't have water inside it. Then it becomes a wet molten globule where every part is hydrated, including the core of the protein, and then you get the fully unfolded state. So this is really telling us that what's important in maintaining the integrity of a protein structure are the van der Waals interactions and hydrogen bonding interactions which keep the structure together. Because when this is happening, the hydrophobic effect is not coming into play at all. So these were equilibrium experiments. It's just going forward. You just, well, these were kinetic experiments. I talked to you about the swelling. Those were equilibrium experiments. And what we then finally, after, it took nearly 10 years after those equilibrium experiments, kinetics is much harder to do normally, to develop that methodology. So these are rather somewhat complicated experiments to do. And we could show that the protein was swelling and losing structure as it swells, but there are continuous swelling processes involved on two different pathways, and then there are first order transitions from these series of conformations to this series of conformations. So this was something which we couldn't understand using the standard ideas which everybody in the field was using. So that's why we had gone to Deepak Dhar at that time, and together with him, we found that our data were describable by a Rouse-like model to describe the swelling process. But what this really told us was about continuous changes on two different pathways during the unfolding process. So at the same time, in the 90s, we had started working and showing that there are multiple ways by which a protein can fold and unfold by just using simple measurements, using, again, stop flow, but with the circular dichroism as the probe and fluorescence as the probe and mutations to change sequence and changing conditions. And we could show that there were at least two pathways for at least bar star to fold and unfold. Now, remember that the statistical mechanical view was that there are essentially an infinite number of pathways. In no experimental study has anyone been able to show more than 2 or 3 pathways. So it's like you have a mountain range, the only way to come down is by using 2 or 3 passes and not just skiing anywhere you want. If you try anywhere else, you fall and die, okay? So this is what our data was showing, and we started off doing unfolding experiments, and more recently, Sandhya in the lab could show that, it wasn't difficult to show you have multiple pathways, 2 or 3 pathways. The question is, how do these pathways differ from each other? That is really difficult, and it took us 10 years from the time we did the unfolding experiments to do the same in folding, because folding is so much faster, 10 times faster. And we could show that proteins look like they have been assembled from their parts. So if you look at a protein, you can see helix here, beta sheet here, and you can easily imagine these things coming together. And that's what actually does happen. So some structure will form fast on one pathway and slow on another. The same structure will form slow on another pathway. So a helix might form fast on one pathway but slow on another pathway. So things were happening independently of each other. And this work of Sandhya's really gave us some general rules applicable, I think, to all proteins and describing how they fold. So we have shown multiple pathways for 5 or 6 different proteins over the years. And some of them we've got structural data, some of them we have not. It's only Sandhya's work with Monell in which he got really detailed ideas about how these pathways differ from each other. So the next thing I want to talk about I've chosen some topics to talk on based on what excited me a lot when the work was done. The question so what I've been trying to tell you when I talk about a protein swelling during unfolding or contracting while folding, I'm basically saying that the chain is adopting a continuous range of structures in either process. It's not hopping from one to the other. So, which means that it's not what we would call a cooperative process. Now, the general idea in the field in the 90s and also in the 2000s even was that folding is two-state. In other words, you have an unfolded chain which hops over a barrier to the folded. It's like magic. Now, people couldn't detect intermediates, but that was because, unlike us, they were using one or two probes only to look at the process. If you use multiple probes, then the complexity of folding becomes obvious. So we started using hydrogen exchange along with mass spectrometry. Again, I will not describe the method. So you can think about these distributions as distributions of some parameter representing structure. And what you can see out here is that this distribution is just changing continuously over a very long time frame. This is the time frame which the protein takes to unfold when it is put in water, okay, 40 days. If you put it in guanidine, a denaturant like urea, it does it much faster, but now what you can see is that it's not, in the initial parts, it's just gradually changing, but then it starts becoming cooperative. In fact, so what's happening, the distribution of molecules, there are two distributions, one comes down and the other goes up. Okay? It's not one distribution of molecules changing continuously. That happens early on, but later one goes down and the other goes up. That's what you would call an all or none transition. And this is something which we couldn't understand what's happening, but because I was teaching my physical biochemistry course and teaching about Marcus's work on electron transfer, it had nothing to do with protein folding, but in one of his reviews, he was talking about how energy barriers change during electron transfer processes. And what you can see quite simply is that if you change relative stabilities, barriers will disappear. Here you have a barrier. You have to go from here to here, you have to climb up and then come down. But here there's no barrier between U and N. Here there's no barrier between U and N. Okay? And that happens just because of change of relative stabilities, as you can see in the manner I've drawn it. Okay? And that's exactly what's happening. When you change, when you add in a little bit of guanidine, you are stabilizing the U state. So you go from this type of scenario to this type of scenario. Okay? And we could then show this is true by doing different things. You could destabilize a protein, you could stabilize the native state, you could add co-solvents which change stability. And everything in respect to cooperativity could be explained on the basis of these structures changing in a gradual, continuous manner. So, so Pooja could then also get detailed structural information and find out why there were pauses during the process. And the very good thing about such work is that you could look at unfolding and folding conditions. And because you could look at unfolding and folding conditions, folding had to be the exact opposite by the law of mass action. And so we knew exactly what was happening during folding also. So I'd like to end by just talking about something which we started working on in the middle of 2000, say around 2005, 2006, which is on protein misfolding and protein aggregation. And so you have, this is the folding process, unfolding, going to the native functional structure, and maybe through some intermediate. But this fellow can start aggregating. You all are familiar with aggregation. When you heat and boil an egg, the protein unfolds and then aggregates. And that white stuff which you eat is aggregated protein. So sometimes when they aggregate, they form very regular structures, which are called amyloid fibrils. And these type of aggregates are associated with many neurodegenerative diseases. So amyloid beta and tau with Alzheimer's, synuclein for Parkinson's, TDP43 for temporal dementia. And the changes in color for this tells you how the disease spreads in the brain. You have aggregates being formed in all cases. The aggregates spread in the brain as shown by the change in color. And so the question is, how do these aggregates spread in the brain? The aggregate has to hop from one cell to the other. And what happens is what Harish showed in the lab a few years ago was the process of, so you have bad protein entering a cell, meeting good protein, and converting the good to bad. It's like an autocatalytic reaction. And he could show that this process is very similar to the well-known mechanism in enzyme catalysis, the Michaelis-Menten mechanism, where enzyme and substrate form a complex and then give you product. Here you have the fibril acting as the aggregate acting as a enzyme, meeting monomer, forming a complex, and everything converting into fibril again. And we could really, this is really very important, I think, because once you have a quantitative way of describing aggregation, you can look at things like inhibitors, etc., which Harish later did. The other problem in protein aggregation, which we've been working on, is the aggregation of the prion protein, where two parts of those subdomains of the protein have to separate for aggregation to happening. And Joginder Singh, a very talented student in the lab, did through a series of experiments, could show that this was actually happening, that two parts of the protein are separating, and because they're separating, that facilitates aggregation and conformational conversion of the protein. And since Suman said nice things about him, I will end up by showing you some work of his, recent work, where he did experiments to show what happens very early on during the misfolding process. He could identify partially unfolded forms which are populated to less than, say, 0.1% and get their structural information on them, and showed what could lead to the separation of those two subdomains. And what he could show is that both perturbing electrostatic interactions at one end of the protein or by perturbing aromatic interactions at the other end of the protein could both lead to separation of these two subdomains from each other. So with that, I'll end the science part, but I want to say a few important things about doing science. One is, it's very important to have good colleagues and collaborators. And I've been fortunate in having these two people around me for most of my career, Panikkar and Matthew. With Matthew, I would have discussions on science virtually every day, Panics perhaps every few days. And both of them really created an environment at NCBS which made it possible to really do good work. Both NCBS and IISER are institutions where I think there's no excuse not to do well. And I think it's because of people like Panikkar and Matthew that it was possible at NCBS. This is PK Maitra, who I mentioned right in the beginning with Vijay. That's me in the background, Veronica. And so he's really the person who inspired me a lot in my early days. The person on the bottom left here is G. Krishnamurthy, who I've collaborated with a lot. His enthusiasm for science was just amazing. And really, I learned a lot from him over the 25 odd years we collaborated. And lastly, there's Krishnan, Richa's thesis advisor. Krishnan, I first met when I went to PK Maitra's lab in that summer of 1978. He would come and talk to me at lunchtime. He would come to Maitra to go to lunch with him, and I was there, and he would ask me questions, science questions. And they were, I thought, pretty straightforward questions, and I would give straightforward answers. And he liked the fact that I could answer his questions. I later learned that these were the questions they asked at the PhD student interviews. But anyway, he liked me a lot because I could answer his questions. He also liked me because he was in one of the first batches of the BRC training school, and my father taught him. So again, it was, I think that also counted a lot to him. So over the years, he was really a source of enthusiasm, a source of advice, a source of, he could discuss anything under the sun. In his postdoc, he had worked on protein folding, so he had some idea about the subject. Of course, he would say much more than what he should, but he had ideas on everything, and to have such a person in an institute or associated institute is really very important. So the other thing about doing science, so it's good to have good colleagues and good collaborators. They're critical for being able to do well. And these are people I've worked with. I've had quite a large number of collaborators. Raghavan was Girish's advisor, has been a close collaborator of mine. We got to know each other at Stanford. He was a PhD student there. And both of us ended up in Bangalore. And it was, I think he probably was on the thesis committees of most of my students out here. There are people both in TIAFA and NCBS who I collaborated with. Matthew was not just a colleague, both of them were colleagues and collaborators. These are more recent collaborators. And I've benefited from my association with all these people on this slide. This is my current set of students. These are students who have, and postdocs who have passed out. So I've been fortunate, and this really is something I'm grateful that I've had the good fortune to have good people to work with over the years. So if you are in a science institute, you invariably have to do non-science work. And all faculty members will complain about doing administrative related work. But it's really very important for doing that. Because if you want the institute to function in the manner you would like it to, each of us has to contribute. And one has to learn to contribute in a manner which doesn't affect your doing the science. And that is something very important. And a lot of us don't want to, do things not connected directly with your research work. That's somewhat selfish. If you help in creating a good environment, your own work will really go well. And so very early on in NCBS, I was just 37, Vijay said I have to become the dean. He didn't really have any choice because I was the first person promoted to associate professor, and so he had no choice but to appoint me as the dean. I remained the dean for 11 years. It was quite a bit of work to be a dean in an institute being set up, but I think it was really very nice finally to shape an institute, to make it function in the manner I thought was the best way for it to function. And Vijay really gave me that freedom to shape things in the manner I thought were important. So that's been very important. So I think, one should carry out administrative duties. One learns from them, one learns how to deal with people, which is very, very difficult. And you finally end up benefiting to a large extent in terms of your own growth as a person. And finally, family support is very, very important. Nathu already referred to this. I was very fortunate, as I said right in the beginning, in having parents who were from an academic background, who understood the importance of hard work and focus, etc., in doing things well. Both my parents lived to a ripe old age, so were there till a few years ago to support in various different ways my career. And my mother was a tough person, and I think both because of that, both my father and I did reasonably well because of her support. she used to say, "Bolne se kya fayda, karke dikhao." And when I was younger, I used to talk much more. And so she, I think, said, "Do things rather than just talk." And I suppose she said the same thing to my father at some stage. And um, I had a sister who passed away when I was just 22 years old. She was a PhD student in the US, got meningitis. And in the early years, the three of them really played an important role in, uh, in my development as a person. Uh, this is Sangeeta, my wife, my two children. At a young age, they're like this, they've transformed into this right now. The reason I put this photo is that their growth as individuals has really paralleled my growth in science in an academic career. And I mean, it's been very, when you're happy at home and these individuals bring joy to you at home, it helps in your work going well. And so I don't know whether they're responsible for my work going well, but certainly there's a parallel, and it's not fully independent processes happening out there. So this is Sangeeta today, again a transformation, a bigger transformation here. But anyway, family support, as I said, is really very important in doing well. So with that, I think I've gone way above time, but thank you for your attention. Thank you for listening to me, and I guess that's it.
We need you on stage.
Huh?
We need you on stage.
Why not?
Thank you, Jayant, for giving such a wonderful presentation and a talk about his scientific achievements. It's a long journey. And so now I'd like to request Professor Bhagwat to come on stage and felicitate, and also Mrs. Udgaonkar, please, to come on stage and to felicitate Jayant. Finally, I'd like to express my thanks to Professor Udgaonkar, Professor Bhagwat, all invited guests and attendees for gracing this occasion. Last but not the least, I would like to acknowledge the organizing committee and supporting staff that helped put together today's event. And so we come to the conclusion of this ceremony. There'll be light refreshments along with tea and coffee that is being served at the foyer right outside this auditorium. Thank you.